Okay, so welcome to this next video in the playlist on lipid modification of proteins. Okay, so in this video what we're going to discuss is the palmitoylation of proteins, and we're also going to discuss an example of an inhibitor of palmitoylation. Okay, so palmitoylation and inhibitors, okay? Right, and the inhibitor we're going to discuss is 2-bromopalmitic acid. Okay, right, so I'm going to start off with a basic discussion of palmitoylation of proteins. We'll then see the main two distinct types of palmitoylation of proteins, and we'll go from there and make our way towards the inhibitors of palmitoylation. The inhibitor that we're going to look at. Okay, so we'll start off with what actually does it mean to palmitoylate a protein. Well, basically what you're going to do is add on palmitic acid molecules. So we need to start off by discussing what a palmitic acid molecule is. Well, palmitic acid also has another name. Its more correct name is hexadecanoic acid. Okay? And although hexadecanoic acid is slightly more of a mouthful than palmitic acid, it's useful because it tells you exactly what you're dealing with here, whereas palmitic acid doesn't really give you any information at all. Hexadecanoic acid tells you that you're dealing with a 16-carbon carboxylic acid. So let's draw out a structure for this. So let's start by drawing out the carboxylic acid group. So here it is. Okay, and now we need 16 carbon atoms. Okay, so we've got one already. Now the other 15 are going to have to follow. So we're going to have 14 methylene groups in the middle. Okay, so here is a methylene group, and because I don't want to have to draw 14 methylene groups out, what I'll do is draw one methylene group, put brackets around it like so, and then subscript this with a 14 to mean repeat this methylene group 14 times. Okay, and then right on the end you then have a methyl group. Okay, so this then is the structure of palmitic acid or hexadecanoic acid. Right, now it's not palmitic acid that you directly add on to proteins. Instead you use something known as palmitoyl coenzyme A, or palmitoyl-CoA for short. So palmitoyl-CoA, and CoA is short for coenzyme A, and this is the same coenzyme A that you will have heard of in the respiratory reactions. Okay, so it's very important there. You have acetyl-CoA all over the place in the respiration reactions. Alright, okay, we won't need the full structure of coenzyme A, so what instead I'll do is I'll draw CoA and put it in a little box like so. We only need to know about one important functional group of coenzyme A, and this is a thiol group, which is a sulfur atom bound to a hydrogen atom. Okay, so whenever you have a sulfur atom bound to a hydrogen atom like this is called a thiol group. And I would urge you to think of thiol groups as being very, very similar to alcohol groups, basically. Sulfur is in the same group of the periodic table as oxygen. So if you were to substitute this sulfur in with an oxygen, that will be an alcohol group there. And as I say, sulfur is in the same group as oxygen, so it has very similar chemical properties. So this indeed is very, very similar to an alcohol group. Alright, so what happens if you want to create palmitoyl coenzyme A? Well, basically, let's imagine for a moment that this isn't a thiol group, but instead it is an alcohol group. So imagine that that sulfur atom is an oxygen atom. Well, you could react this alcohol group on this coenzyme A molecule with the palmitic acid molecule's carboxylic acid group. Okay, what you could do is you could take the alcohol group off of the carboxylic acid group of the palmitic acid molecule, and you could take the hydrogen atom off the what we're imagining is an alcohol group of the coenzyme A molecule. You could combine those two together to make water, okay, and then you could bind this, what we're imagining is an oxygen, to the carbon of the carboxylic acid group. And this would be called an esterification reaction, and the link between the palmitic acid molecule and the coenzyme A molecule would then be called an ester link. Now, of course, we have to revert back to the actual scenario, which is that we have a sulfur atom here, not an oxygen atom. But I said that these thiol groups were very similar to alcohol groups, and in fact, thiol groups can react with carboxylic acid groups in an identical way, pretty much, to alcohol groups would. However, this link now is not called an ester link, but instead is called a thioester link. Okay, so you have a thioester link between palmitic acid here and the coenzyme A molecule. Okay, and that is then called palmitoyl coenzyme A. And I would just like to explain what palmitoyl means. Palmitoyl means this group here. So basically, palmitic acid was the entire molecule when you had the alcohol group on the carboxylic acid group. But as you can see, we're not adding the entire palmitic acid molecule onto the coenzyme A molecule. Instead, we're just adding this group that I've now circled in green without the alcohol group of the carboxylic acid group. Okay, and this group here that I've circled in green is called the palmitoyl group, and that's why this molecule is called palmitoyl-CoA, because effectively we've just taken the palmitoyl group and bound it to coenzyme A. Okay, right, so let's now discuss palmitoylation of proteins, and it's going to involve this substrate here, palmitoyl coenzyme A, because we're not just going to take a palmitic acid molecule and add it onto proteins. Instead, we're going to use palmitoyl coenzyme A, and we'll transfer the palmitoyl group off of the coenzyme A molecule and onto the protein. Okay, so we're going to start off with a very rare form of palmitoylation, rare compared to the other form, okay, which is N-palmitoylation. Now, this isn't really the star of the show. S-palmitoylation, which we're about to come to, is the star of the show, but for completeness, I want to mention N-palmitoylation. Okay, right, so what is N-palmitoylation then? So, basically, if you have a protein, which I'll just draw like this, so remember, proteins are all polypeptides. They're polymers of amino acids, so it's amino acid after amino acid after amino acid after amino acid. Okay, so let's say that we have here the first amino acid of the polypeptide. Well, the first amino acid has a very special group. It has a free amino group, basically, because every other amino acid in this polypeptide, so for instance, if we just take this one in the middle, will have its amino group bound to the carboxylic acid group of the amino acid in front of it, basically. So it will not have a free amino group. Its amino group will be involved in the peptide link. Okay, so this first amino acid, number 1 over here, is the only amino acid to have its free amino group. Similarly, the last amino acid over here is the only amino acid to have a free carboxylic acid group, because all other amino acids in the polypeptide have their carboxylic acid group linked to the amino group of the next amino acid along, basically. Okay, so basically, N-palmitoylation can occur to proteins which have a cysteine residue at position 1. Okay, so let me show you the structure of a cysteine amino acid at position 1. So here is this free amino group here, and then you have an alpha carbon following it with a hydrogen coming off it, so this is just core amino acid structure here. Here's the carboxylic acid group of the cysteine amino acid, which will be linked to the amino group of the next amino acid along. And the R group of the cysteine amino acid consists of a methylene group, like so, with a thiol group coming off it. Okay, right, so if you have cysteine residues in position 1 on your polypeptide, you can perform N-palmitoylation of the polypeptide, and you're going to basically add the palmitoyl group onto this amino group here, which is why it's called N-palmitoylation, because you're going to attach it to the nitrogen atom here. Now, I just want to clarify something that you might be wondering. Basically, you might be wondering, well, how on earth do you get a cysteine amino acid in position 1? Because I know something about the first amino acid in polypeptides. it's always the same amino acid, because remember, in the process of translation, so let me just draw a little ribosome here. Okay, so in the process of translation, what happens is the mRNA is here, and to begin the process of translation, you have a start codon, which is let me see if I can fit this in, UG, which is quite helpful because you can think of it as augment, being short for augment, basically, which means get bigger. So it's kind of fitting that the start codon should be AUG. And this start codon always codes for the same amino acid, and that same amino acid is a methionine. Okay, so you always end up with a methionine in position 1. So how on earth would you get a cysteine amino acid in position 1? Well, methionine is what's called the initiator amino acid. Okay, so indeed, when you first produce a protein, you will always have methionine in position 1. It is the initiator amino acid. However, you don't have to keep it there. You can sort of chop it off afterwards, and then the second amino acid along will become the first amino acid. And if that second amino acid is a cysteine residue, then you'll have a cysteine in position 1. So basically, methionines, these initiator amino acids, are often cut off, basically. So that's how you can end up with proteins that don't have methionine in position 1. Okay, so if you have a cysteine in position 1, then you can undergo NParmittylation, but let me stress this, it's only if you have a cysteine in position 1. If you've got any other amino acid, forget it, basically. It's cysteine, cysteine, cysteine. Okay, so what will happen is you'll bring in the palmitoyl-CoA molecule. So let me show this here. So here is our palmitoyl-CoA molecule. Here's coenzyme A, linked via this thioester link to the palmitic acid molecule, which remember has these 14 methylene groups. So here's one, I'll bracket it and put 14 there, and then a methyl group on the end. So this is palmitoyl coenzyme A coming in over here. Okay, and what happens is you will break this bond between the carbon and the sulfur atom, and remember, covalent bonds contain 2 electrons, 1 electron from each component of the bond. So there'll be 1 electron from the sulfur atom and 1 from the carbon atom. So for the basis of understanding this reaction, and not in any way, shape or form intended to be an electronic flow diagram, you can imagine that you're giving the electron from the carbon back to the carbon and the electron from the sulfur back to the sulfur. Of course, that is not what will actually happen when this chemical reaction occurs. Homolytic fission leads to free radical chain reactions, okay? So it's not something that will be occurring here, okay? But for the basis of understanding, it's a helpful simplification. Okay, and again, we're cutting the bond between this nitrogen and this hydrogen. Again, imagine giving 1 electron back to the nitrogen and 1 back to the hydrogen. What you can then imagine doing is binding this nitrogen to the carbon atom over here of the palmitic acid molecule, or of the palmitoyl group, if you like, because after all, it's no longer palmitic acid, because where's the alcohol group? Okay, and of course we can do that because the nitrogen had a free electron and the carbon also had a free electron. Then you can imagine forming a bond between this sulfur and this hydrogen, both of which again have free electrons, and that will recreate you the coenzyme A molecule. Okay, right, and now what you have is this amide link between the carboxylic acid group of the palmitic acid molecule and the amino group of the first cysteine in the polypeptide. And this is why this is called NParmittylation, because the palmitoyl group over here is attached to the nitrogen atom of the first amino acid along. Okay, so that's NParmittylation, and that is quite rare, basically. It's not a common way to palmitylate proteins. Okay, so we're not really going to discuss NParmittylation anymore. It's catalyzed by a discrete set of enzymes that are different from the enzymes which catalyze what we're turning onto now, which is called S-palmitylation. And this is far more important, basically. Okay, so S-palmitylation. Right, Okay, so let me firstly give an introduction to S-palmitoylation, and then I'm going to tell you about the 4 different types of S-palmitoylation, and they all follow the same pattern. It's just basically trying to explain when S-palmitoylation occurs. So really, the 4 different types are actually 4 different examples of when it occurs, when it would occur, basically. So, S-palmitoylation will occur again on cysteine residues. Basically, this is the running theme. You can only add palmitoyl groups onto cysteine residues, no other amino acid. Okay, so S-palmitoylation, however, is not going to occur onto cysteine residues that are in position 1. Instead, it's going to occur on cysteine residues that are in the middle of the polypeptide, and I should have written cysteine's name at some point. So this is the amino acid cysteine here. Okay, and the single letter amino acid code for cysteine is C. Right, so let me show you what happens in S-palmitoylation. So I'll show you another cysteine residue, and then I'll show you when it happens. Well, the 4 different cases that are known about when it actually happens. Okay, and then we'll discuss the enzymes which catalyze this. Right, so here is the core amino acid structure, and notice that now we've got the amino group and the carboxylic acid group involved in peptide links to neighbouring amino acids. And the R group of cysteine, once again, is a methylene group with a thiol group coming off it. Okay, and now let's bring in the palmitoyl-CoA molecules. So here is coenzyme A, with its thiol group that's linked via a thioester link to the palmitic acid molecule here. Okay. And of course we have 14 methylene groups, so I'll draw 1, bracket it and put a 14 there, and here's the methyl group coming off there. Now, this time what's going to happen is we're going to transfer this palmitoyl group onto that thiol group there. So notice we have another thiol group here on our cysteine residue, and basically what we're going to do is transfer the palmitoyl group from the thiol group of the coenzyme A molecule onto the thiol group of the cysteine residue. Right, and that's why it's called S-palmitoylation, because we're adding it onto a sulfur atom. Okay, so again, imagine cutting this bond between the carbon and the sulfur atom of the palmitoyl-CoA molecule. And again, imagine cutting this bond between the sulfur and the hydrogen atom of the thiol group of the cysteine residue. And again, imagine 1 electron goes back to each component. Then we can link this sulfur atom, to this carbon atom over here, because both of them have 3 electrons, and we can link this hydrogen onto the sulfur atom of the coenzyme A molecule to recreate coenzyme A. And that will then create palmitoylated cysteine. Okay, an S-palmitoylated cysteine residue. Right, okay, so what we now want to turn our attention to is the 4 different times that this would happen, because you know, if you've got a massive great polypeptide, you could have a huge number of cysteine residues within it. Are you going to palmitoylate every single cysteine residue within that protein? Well, the answer is of course no. You're only going to palmitoylate a few of them. So we now want to see what dictates whether a cysteine gets palmitoylated or not. When do cysteines get palmitoylated? Because we have a huge number of them, and you're not going to palmitoylate all of them, so there must be some sort of selection criteria. And I'm now going to tell you about 4 different scenarios in which a cysteine residue would get palmitoylated, but we'll do that in the next video.